Good day, everyone. For today's topic, we'll be discussing about the regulation of gene expression. So the following are the references of this lecture. Now, let's have this question before we start our main discussion. Why do we have different types of body tissues? So this is quite a fundamental question in anatomy and physiology and perhaps in histology. But the usual answer for this question is because the different body tissues have different body functions, therefore they need to adapt to a certain function of an organ system, and that's why they have body tissues. But the fundamental and most molecular reason why we have different types of body tissue is because several genes are only expressed on a certain type of cell. Now, not all genes are expressed in our DNA. It is selective and it is governed by the regulation of gene expression. Now, every cell has the same DNA and therefore contains the same genes. So basically, all cells in our body, so it can be from our bone cells, from the liver, the muscle, from our spermatozoa, the embryo, they have as quite similar genetic makeup, genetic component, but different genes need to be on and off in different types of cell in order to have a specific differentiated type of cell. Now, therefore, gene expression must be regulated. And this is the reason why we have different types of body tissues, why we have um, different types of phenotype of a certain characteristic. It's because of the regulation of gene expression. The gene expression must be regulated in several different dimensions from the moment that we are conceived and from the moment that we develop into a child and eventually to adults. Now, with respect to nutrition, gene regulation is important to guide development of organs, tissues, and cell types required to ingest, digest, and metabolize nutrients. So the reason why we have different types of functions in our body is because several genes are regulated, several genes may be expressed or may be repressed. And expression of certain genes can be expression of enzymes. And these enzymes can help in nutrition, in the development. It can be for structure or structural. So that's how important gene regulation is. Now, before understanding about gene regulation or how gene expression is regulated, you have to first review on what is gene expression. So the cellular concentration of a protein is determined by a delicate balance of at least 7 processes, each having several potential points of regulation. Now we have this gene expression, the process of gene expression, or the central dogma of molecular biology. Okay, but if you focus more on the gene expression, we have these processes. First is the synthesis of the primary RNA transcript. This is what we call transcription. Next is post-transcriptional modification of mRNA. Once the mRNA is produced or is synthesized, that RNA can undergo processing. Now we have also messenger RNA degradation, which is quite a form of post-transcriptional processing also. Then we have protein synthesis or translation. In the process of protein synthesis, the mRNA is read by the ribosome to form protein. And once proteins are formed, these proteins can be modified through the process of post-translational modification of proteins. The next, we have protein targeting and transport. And lastly is protein degradation. Now, all of these processes during gene expression can be regulated at a certain point. And it can also affect expression of genes or expression of a certain product of the gene. So again, to further illustrate about gene expression, now the conversion of or or the synthesis of RNA from the DNA template is what we call transcription. And that transcription is also a point of regulation. So we have also here for initiation of transcription, that transcription can be activated or repressed. You can see later after formation of the mRNA. Okay, so the mRNA can be further modified through the process of RNA processing. So we have capping, cleavage, polyadenylation, also splicing. It is a form of post-transcriptional modification. Now, when the mRNA is produced, it can leave out of the nucleus. If it's a eukaryotic cell, it can leave out from a nucleus and undergo the process of translation. And in this process, translation can also be controlled. And once translation is already done, forming the protein, can now undergo post-translational modification. Now let's move on to the principles of gene regulation. The controlling gene expression is often accomplished by controlling transcription initiation. So this is one of the most common way of controlling gene expression. It's actually the process of transcription initiation, the start of transcription, or the start of synthesizing RNA from DNA. The control of transcription ultimately results from changes in the mode of interaction of specific regulatory molecules, usually proteins, okay, with various regions of DNA in the regulated gene. Now we have also regulatory proteins. Now these regulatory proteins bind to DNA to either block or stimulate transcription, depending on how they interact with the RNA polymerase. Now remember, RNA polymerase is important for transcription because RNA polymerase synthesizes RNA. So this is the basic principle. Transcription, or the process of reading the DNA template to form the RNA transcript, is mediated and regulated by protein DNA interactions, especially those involving the protein components of RNA polymerase. The controlling gene expression, again, this is just a repetition of the previous slide. Now we have housekeeping genes. Now what are housekeeping genes? Now housekeeping genes, these are genes for products that are required at all times, okay, which are expressed to a more or less constant level virtually every cell of a species or organism. So from the name itself, housekeeping, it should be there. These genes should be expressed almost at all times. Okay, just like in your home, your home, in order for it to be a healthy home, your home should be the every now and then should be tidy, should be cleaned up. Okay, so you need to do housekeeping to make your home healthy, functional, nice place to live in. So in our cells, it also have its own housekeeping proteins that is uh, encoded by the housekeeping genes. Now, when we say constitutive gene expression, this means unvarying expression of a gene, meaning a gene is always expressed. When you say unvarying, it does not quite vary, meaning it is consistent, meaning gene expression is almost always present. So that is constitutive gene expression. When we say regulated gene expression, the gene products of regulated gene expression, which cellular levels rise and fall in response to molecular signal. That is why it's called regulated, because gene products are formed when there are several molecular signals. So gene expression can be turned off or turned on depending on the type of signals that are present. Okay, so when you say constitutive gene expression, the gene is always expressed to form that protein. Now, for regulated gene expression, the expression of the gene can be turned off or turned on. Now we have also these terms. When you say inducible, it refers to gene products that increase in concentration under a particular molecular circumstance. So these are gene products that increase in concentration. When you say induction, it is the process of increasing gene expression. When you say repressible, this refers to gene products that decrease in concentration in response to a molecular signal. When we say repression, it refers to the process of increasing gene expression. So prokaryotic organisms regulate gene expression in response to their environment because of their simple structure. Usually they respond to whatever signal that they have in the environment. Can be through nutritional, can be through presence of molecular signals. So that is how prokaryotic organisms generally control gene expression. For eukaryotic organisms, since eukaryotic organisms are quite complex, they rely gene expression regulation to maintain homeostasis inside the organism. So the gene regulation process is equipped or is geared towards maintaining balance within the system of that organism. So that is um, the basic orientation of gene expression regulation in eukaryotic organisms, especially mammalians and humans as well. Now, we have several types of gene regulation. We have positive regulation and negative regulation. When you say positive regulation, this is the expression of genetic information that is quantitatively increased by the presence of a specific regulatory element. The element or molecule mediating positive regulation is what we call a positive regulator or activator. So again, when we say positive regulation, the expression of genetic information is increased by, a, by the presence of a specific regulatory element or a specific molecule or a protein that helps boost expression of the genes. When you say negative regulation, it's the opposite. The expression of genetic information is diminished. The term here is diminished by the presence of a specific regulatory element. The element or molecule mediating negative regulation is said to be a negative regulator or repressor. Now, again, the element or molecule that mediates positive regulation is what we call an activator. For negative regulation is what we call the repressor. Now, double negative has the effect of acting as positive. So if you have two repressors, most likely it will result into a positive regulation. So an effector that inhibits the function of a negative regulator will appear to bring about a positive regulation. Because if you inhibit an inhibitor, if you inhibit an inhibitor, okay, for example, if molecule A is an inhibitor and molecule B is an inhibitor, then knowing from the information that molecule A inhibits molecule B, okay, the overall process will be negated or will be neutralized. So the whole process will be of positive regulation because you actually inhibit the inhibitor, which makes it activate the genes. So we have types of responses to a regulatory signal. Now, gene expression regulation involves signals. It will signal the genes to express or not express. Now we have several behavioral types of responses to regulatory signal, and one of it is type A. We have three types, type A, B, and C. For type A, the increased rate of gene expression is dependent upon the continued presence of the inducing signal. Now, gene expression will only occur if there are signals present. So, once the inducing signal is removed, the gene expression diminishes to its basal level. And if the signal is again present, the rate of gene expression increases. The example for this one is prokaryotes. For in prokaryotes, it is exemplified in response to a sudden change sudden change of the intracellular concentration of a nutrient. For eukaryotes, after exposure to inducers such as hormones, nutrients, and growth factors, it can also alter gene expression. So if you have hormones that causes increase in gene expression, it will only occur if that hormone is present. If that hormone is not present, then gene expression will not occur or will be diminished. For type B response, the rate of gene expression is transient. Okay, the rate of gene expression is transient even in the continued presence of the regulatory signal. So we have response desensitization and response and recovery phases. It commonly occurs during the development of organism. So for type B response, you have a signal that is quite persistent. Okay, if there's a present phase signal, but gene expression is transient, meaning it only occurs a few times, okay, or for a moment only, amidst the presence of the signal. Okay, for type C, there is an increased rate of gene expression that persists indefinitely, even after termination of signal. So if you have a signal that is only present even for a short period of time, the gene expression persists indefinitely. There's an increased gene expression indefinitely. So the signal can act as a trigger, and this occurs during differentiated function in a tissue or organ. So this is one type. Now let's discuss first about the anatomy of a gene. What can we see in a gene? So remember from the lectures previously about introns and exons. So we have introns and exons. The exons are the ones that code for a certain protein. Now, for a DNA, we have a control region and a coding regions. Okay, in the control regions, so let's have first the coding regions. The coding regions contains introns and exons. Now these exons are the ones that are um, usually transcribed to form protein, and we have the control regions. This control region contains several regions. We have promoter and enhancer regions. So later on, we'll be discussing specific examples of promoters and enhancer regions of genes. So for the regulation of transcription initiation, so this is what I've previously mentioned. This is one of the most common way of how genes are expressed is through the regulation of transcription initiation or regulation of the starting of transcription. So there are at least three types of proteins that regulate transcription initiation by the RNA polymerase. We have these specificity factors, the repressors, and activators. When you say specificity factors, they are usually proteins that alter the specificity of RNA polymerase for a given promoter or set of promoters. Example of which is the sigma subunit of the E. coli polymerase holoenzyme. Okay, so this is a specificity factor. It mediates promoter recognition and binding. The next type of proteins that regulate initiation of transcription are the repressors. They are the one that impedes access of RNA polymerase to the promoter. So remember from the discussion on transcription, in order for the RNA polymerase to transcribe from RNA, it needs to bind to the promoter. Okay, we have activators. The activators, they enhance the RNA polymerase promoter interaction. Binding sites are what we call operators. Okay, wait. Um, Regulation in prokaryotes and eukaryotes are quite different. So I'm just gonna give you a short overview, but we will deal more deeper discussion this one in the later slides. But in general, in prokaryotes, the binding sites are what we call operators, and they are generally near a promoter. When a repressor is present, the RNA polymerase binding or movement along the DNA after binding is blocked. So if there's a repressor, RNA is not bound to the operators. When you have negative regulation, this is regulation by means of a repressor protein that blocks transcription. Once a repressor binding to DNA is regulated by a molecular signal, usually a small molecule of proteins that binds to the repressor, it causes a conformational change. And the interaction between the repressor and the signal molecule either decreases or increases transcription. It actually depends on the signal. For eukaryotic cells, the binding site for a repressor may be some distance from the promoter, and binding of this repressor to their binding site has the same effect as in bacterial cells. They inhibit assembly or activity of a transcription complex at the promoter. Now remember, in prokaryotes, the genetic architecture is quite complex compared to prokaryotes. So we have common patterns of regulation of transcription initiation. So again, you have negative regulation. So for this one, a repressor binds to the operator in the absence of a molecular signal. The external signal causes dissociation of repressor to permit transcription. Now, please take note that the positive and negative regulation, they refer to the type of a regulatory protein involved. The bound protein either facilitates or inhibits transcription. So in either case, the addition of the molecular signal may increase or decrease the transcription depending on its effect on the regulatory protein. So for negative regulation, okay, the molecular signal causes dissociation of repressor from DNA, inducing transcription. Even if it causes transcription, the signal by which it binds to the repressor is negative, meaning it is negative to the repressor. So based from the principle that we have learned before in the previous slide, there are double negatives, it forms a positive response. So for this one, once a repressor that binds to the operator in the absence of a molecular signal, the gene is off. Now, if an external molecular signal is present and that signal binds to the repressor, causing dissociation of repressor from the operator, it causes gene expression. So this is also a type of negative regulation. So another type of negative regulation is this one. A repressor binds in the presence of a signal, and that signal causes binding to the operator. And that binding of the repressor to the operator turns off the gene expression. Okay? Again, to differentiate from this one to this, both of them are negative regulation. Now, what's the difference? Is that in this one, in the first example, the binding causes dissociation of the repressor, causing expression of genes. For this type of negative regulation, the molecular signal binds to the repressor, okay, and this causes turning off of the gene regulation. The next is positive regulation. For positive regulation, an activator is present. Okay, so again, to make yourself not confused with the terminologies. When you say negative, it refers to this type of protein. It refers to the protein involved. Okay, to the regulatory proteins that are involved. For both of A and B, we have repressor proteins. So basically, both of these examples or patterns, they undergo negative regulation because again, the protein that is involved is a repressor. If the protein that is involved is a repressor, that signal or that pattern is a negative regulation. For this positive regulation, we say that it is positive because the protein involved is an activator. Okay, so for this one, an activator binds in the absence of the molecular signal and transcription proceeds. When a signal is added, it binds to the activator, it dissociates from the RNA polymerase, causing turning off of the RNA polymerase. So for this one, the RNA polymerase needs an activator in order for it to be active. But if there's a molecular signals that inhibit the activator, therefore RNA polymerase will not also be activated. So even if the function of this one is to the presence of a molecular signal causes turning off of genes, this is still what we call a positive regulation because we have an activator that is involved. Next is another possibility is positive regulation, wherein the molecular signal activates the activator. If we activate the activator, that activator will in turn activate the RNA polymerase to proceed to gene expression. For eukaryotic transcription regulation, the distance between binding sites of activators or repressors and promoters is bridged by looping out the DNA in between. So there's a quite far distance from the coding region and the control region. So they would in transcription regulation there will be usually looping of the DNA. So the looping is facilitated by proteins called architectural regulators that binds to intervening sites and facilitate the looping of DNA. The most eukaryotic systems involve protein activators. Okay, the actual interaction between the activators and the RNA polymerase at the promoter is often mediated by intermediary proteins called activators or called co-activators. Now, activators, wait, no, that's a defective slide. So we have also regulatory proteins. The gene expression is often controlled by regulatory proteins binding to specific DNA sequences. The regulatory proteins gain access to the bases of DNA at the major groove, and the regulatory proteins possesses DNA binding motifs. So again, regulatory proteins, they bind to the major groove of the DNA. To bind specifically to DNA sequences, regulatory proteins must recognize surface features on the DNA. Now, most of the chemical groups that differ among the four bases and thus permit discrimination between base pairs are the hydrogen bond donor and acceptor groups that are exposed in the major groove of DNA, and most of the DNA protein contacts that impart specificity are hydrogen bonds. Okay? The protein DNA contacts are also possible in the minor groove of the DNA, but majority again of protein DNA contacts. So for this one, from how a protein binds to a DNA, it's actually mostly in the major group, but it is also possible that protein can bind to DNA in the minor groups of DNA, but the hydrogen bonding patterns have generally here in the minor groups generally do not allow ready discrimination between the base pairs. So within the regulatory proteins, so for the proteins that binds to the DNA, remember that proteins are made up of amino acids. Now the amino acid side chains most often form hydrogen bonding to the bases in the DNA. And these are the asparagine, glutamine, glutamate, lysine, and arginine residues. Okay, just to have a short review of the major and the minor groove. So this is the major groove and this is the minor groove. Now, as you can see from their molecular structure, we have several atoms that are available for hydrogen bonding. And this is what is very important when proteins bind to the specific DNA molecule. So for example, RNA polymerases or repressors or activators, they usually bind to this part, the major groove, this one, major groove, this is the major groove, and the usual intermolecular forces that involve the binding is due to the hydrogen bonding of the side chains of the amino acids. Again, it's the side chains of the amino acids of the proteins. Okay. Now, we have specific amino acid residues. So, for example, for this one, for glutamine and arginine, these are components of proteins, right? So through their side chain, it forms hydrogen bonding with the nucleobases Okay, they form hydrogen bonding with the base group. Now, several motifs compose the DNA binding domains of regulatory transcription factor proteins. Now, DNA binding motifs are regions of regulatory proteins that bind to DNA. So we have also regulatory proteins that have protein-to-protein interactions. For this one, this is a DNA to protein. This one is protein to protein. So first is we have the DNA binding motifs. And we have three types. We have the helix-turn helix motif, the zinc finger motif, and homeodomain. For proteins that have also protein-to-protein interaction, we have leucine zipper motif and the basic helix loop helix. So generalizations regarding DNA binding motifs. So this is very important. So these are basic principles. Binding of the motifs must be of high affinity to the specific site and of low affinity to other DNA. Small regions of the protein make direct contact with the DNA, and the rest of the protein, in addition to providing the transactivation of domains, may be involved in the dimerization of monomers of the binding protein and may provide a contact surface for the formation of heterodimers and may provide one or more ligand binding site or may provide surface for interaction with co-activators, co-repressors, or transcription machinery. The protein-DNA interaction made by these proteins are usually maintained by hydrogen bonding, ionic interaction, and van der Waals forces. The motifs found in these proteins are class-specific. The presence in a protein of unknown function suggests that protein may bind to DNA. Proteins with a helix-turn helix or leucine zipper motifs, they form dimers, and their respective DNA binding sites are symmetric palindromes. Meaning, if you read that binding sites in reverse, they are in palindrome, meaning they are usually mirror images. In proteins with a zinc finger motif, the binding site is repeated 2 to 9 times. And these features allow for cooperative interaction between binding site and enhance the degree of affinity and binding. So what are examples of transcription factors that contains various DNA motifs? So we have this type of motif, the helix-turn helix, the zinc finger, and leucine zipper. So please remember this table, and I want you to memorize this because these are examples of the several binding motifs. So for helix-turn helix, examples of proteins, if it's from the E. coli, we have the Lac repressor, which is involved in the Lac operon, and the CAP, which is also involved in the Lac operon. For Fudge, we have the lambda CL, the CRO, and the 434 repressors. For mammalian, we have the homeobox protein PIT1, OCT1, and OCT2, which are involved in embryogenesis. For zinc fingers, for E. coli, we have gene 32 protein. For yeast, we have the GAL4. For the Drosophila, we have the serendipity protein and hunchback proteins. For the Xenopus, we have the transcription factor 3A. For mammals, we have the steroid receptor family, SP1. For leucine zippers, for yeast, we have the GCN4 protein. And for mammals, we have the CEBP, FOS, June, FRA1, CRE, binding protein of the CREB, the CY, CMYC, NMYC, and LMYC. Okay, so these are examples of the transcription factors. They are generally transcription factors. They affect transcription, and they have this type of motif, or they have this type of motif or their protein structure. Okay. Well, now let's discuss each by each the types of motifs. So the first type of motif that is about to discuss is the helix-turn helix motif. So this is one of the motifs that are common in prokaryotes. So the helix-turn helix motif comprises about 20 amino acids in the two short alpha helical segments. So we have alpha helical segments, each 7 to 9 amino acid residues long. So each alpha helix, okay, they have 7 to 9 amino acid residues, and they are separated by a beta turn. Now remember, beta turn, this is not a beta pleated sheet. This is a turn. Okay. So for alpha, so the helix turn helix proteins, so again, it consists of a alpha helix, and that alpha helix, it is connected to a beta turn. Okay, example again of the helix turn helix motif, we have the tryptophan repressor, the lambda crow, which is found in bacteriophages, with the lambda repressor fragment, the CAP fragments. The next type of motif to be discussed is zinc fingers. So in a zinc finger, there are about 30 amino acid residues that form an elongated loop held together at the base by a single zinc ion, which is coordinated to of the residues. Can be 4 cysteine residues or 2 cysteines and 2 histidines. So for this example, that zinc is coordinated by 2 cysteines and 2 histidines. It's called a zinc finger motif because we have a presence of a zinc that are coordinated with the 4 different types of amino acids. The protein containing zinc fingers appears to lie on one face of the DNA helix with successive fingers, alternatively positioned in one turn in the major groove. Now, please remember that the zinc itself, the zinc ion, it does not interact with the DNA. Rather, the coordination of zinc with the amino acid residues stabilizes this small structural motif. Okay, so basically the function of that zinc is to stabilize the motif. Now, we have several hydrophobic side chains in the core of the structure, and it also lends stability. Now, many eukaryotic DNA binding proteins contain zinc fingers, and the interaction of a single zinc finger with DNA is typically weak, and many DNA binding proteins have multiple zinc fingers that substantially enhance binding by interaction simultaneously with the DNA. So again, this is the usual structure of the zinc finger motif. It's common in eukaryotes. Okay, so we have several examples of zinc finger motifs. Next is we have the homeodomain. So for homeodomains, it consists of domain of 60 amino acids. The DNA binding segment of the domain is related to the helix turn helix motif. So when you hear homeodomains, we have a somewhat similar but not equivalent. We have a similar um, appearance or a structure to the helix turn helix motif. The DNA sequence that encodes this domain is called the homeobox. Okay, so the DNA that forms this homeodomain is what we call homeobox, and it is identified in some proteins that function as transcriptional regulation, especially during eukaryotic development. Next, we have the leucine zipper motif. Now, this motif is an amphipathic alpha helix with a series of hydrophobic amino acid residues concentrated on one side with a hydrophobic surface, forming an area of contact between the two polypeptides of a dimer. Okay, wait for a while. Okay, now just to review, when you say amphipathic, it refers to a property of a molecule or a substance having both hydrophilic and hydrophobic parts. So you would expect that for leucine zipper, you need to see where the hydrophobic and the hydrophilic part. Okay, so again, this is an amphipathic. So you need to expect a hydrophobic and a hydrophilic. So a striking feature of these helices is that the occurrence of leucine residues at every, remember this, 7th position. So the leucine residue is present at every 7th position, forming a straight line along the hydrophobic surface. So this is the usual structure of the leucine zipper. Okay, you have this hydrophobic component, and this portion wherein it binds, or the DNA binding portion of the leucine zipper, is basic. Okay? It contains a lot of basic amino acids and it has the capacity to bind with the DNA. Okay, so the DNA binding domain of leucine zipper contains a high concentration of basic amino acid residues such as lysine or arginine residues, specifically lysine or arginine residues that can interact with the negatively charged phosphates of the DNA backbone. So remember that lysine and arginine, they have a positively charged side chains. And remember also from that in the DNA, the phosphate backbone of the DNA consists of a negatively charged phosphate because of the oxygen atoms. So a positive and negative charge causes this electrostatic interaction to occur in this portion of the DNA binding region, so which makes this part relatively hydrophilic compared to this one. That's why this leucine zipper motif is an amphipathic alpha helices. Okay, so for now the leucine, so for the hydrophobic region, the hydrophobic region contains leucine, okay, that face each other. So although researchers initially thought the leucine residues are interdigitated, hence the name zipper, we now know that they line up side to side as interacting helices coil around each other, forming a coiled coil. So the original concept of the leucine zipper is that the leucines are interdigitated towards each other, meaning they are windowing. That's the original concept. But now, the current model for the leucine zipper is that they are lined up side to side. So this is in virtue of the hydrophobic interactions. Remember, leucine is a hydrophobic amino acid. So they can easily bind up together because of hydrophobic interaction. Okay. So again, to show you that the leucine occurs almost 7. So 1, 2, 3, 4, 5, 6, 7. It occurs on the 7th amino acid. So 1, 2, 3, 4, 5, 6, 7. Okay, 1, 2, 3, 4, 5, 6, 7. So the leucine zipper uh, contains leucine in every 7th amino acid residue. Next, we have the basic helix loop helix. So the basic loop helix loop helix occurs in some eukaryotic regulatory proteins implicated in the control of gene expression during the development of multicellular organism. Usually, they are um, present in proteins that are involved in embryogenesis and in development. So these proteins share a conserved region of about 50 amino acid residues important to both DNA binding and protein dimerization. And this region can form two amphipathic helices linked by a loop of variable length. Note, now this is helix loop helix is different from the helix turn helix. So this one is a helix loop helix. There's a loop containing amino acids also. So it forms by association of four amphipathic alpha helices to form a bundle. So the basic domains of each monomers protrude into the major grooves on the opposite side chains. Again, the basic portion containing the amino acids, the basic amino acid, they usually have a positively charged and they electrostatically form a bonding with the negative phosphate group. So that is why it causes binding to the DNA molecule. Examples of the basic helix loop helix are the MyoD family, which is involved in muscle differentiation, and the E proteins. These E proteins are required for the development of some white blood cells. Now let's discuss about the prokaryotic gene regulation. The bacteria often respond to environmental change by regulating transcription. Again, bacteria often respond to the environment or changes in the environment, whether it's nutritional, temperature, or motion. Okay, they can regulate gene expression by regulating transcription. The natural selection has favored bacteria that produce only the products needed by that cell. A cell can regulate the production of genes by feedback inhibition or by gene regulation. Now, gene expression in bacteria is controlled by the operon model. Okay. Now, further discussing about prokaryotic regulation, specifically on transcription initiation control, it can be positive control and negative control based again on our principle that when we say positive or positively regulated, there are presence of activators. When we say negatively controlled, we have presence of repressors. Okay. Now, prokaryotic cells often respond again to, okay, I think I've discussed this one. So let's now, before dealing with the specific operons, let us describe what is an operon. Okay, an operon is an entire stretch of DNA that includes the operator, the promoter, and the genes that they control. Okay, so this is the basic structure of an operon. So for this, again, components of the operon, we have the promoter, we have the operator, and the and genes transcribed as unit. Now we have regulatory sequences that are present. So we have also activator binding site and repressor binding site. Now repressors usually bind to the operator, and activators, they are usually found upstream towards the promoter, okay, upstream from the promoter. Now repressor, they usually bind to the operator. And the genes, they are basically genes, they are the one that are transcribed to form proteins of a certain molecule or for protein molecules such as enzymes or structural proteins, not for that matter. Now we have the first type of operon, which is an induction type. So we have an induction type and a repressor type or repressor system. So we have an induction system and a repressor system or repression system. So for induction system, the best example for this one and the most tested one is the lac operon. For the lac operon, the system is initially off. Okay? The presence of an inducer, usually a substrate that needs to be broken down, turns it on. The inducer binds to the repressor, makes it inactive, so transcription can occur. So a presence of an inducer, for example, in the lac operon, the inducer for lac operon is lactose. It binds to the repressor, and that repressor, once that inducer binds to the repressor, it makes it inactive. So it causes transcription of the genes involved in the lac operon. So the inducer acts an allosteric effector and changes the shape of the repressors. Example of which again is the lac operon, the lactose operon. Now before dissecting further about the lac operon, we need to describe what is the role of lactose in prokaryotes. Why are we discussing about lactose and how lactose is normally metabolized in bacteria? Now we have several proteins that are involved. So we have the permeases, we have the beta-galactosidases, and the transacetylases. Okay? For permease, or also known as the lactose or galactoside permease, okay, so you see this one, you see this? This is a protein, right? So this is the cell of the bacteria. So this is the protoplasm, this is the cell membrane, this is the extracellular space. So the first key protein involved is the galactoside or the lactose permease. So that lactose permease, this is a transmembrane symporter. Basically, it's a protein that is found in the plasma membrane of the bacteria that functions to pump beta-galactosides such as lactose into the cell using a proton gradient in the same direction. So basically, the role of that permease is to bring lactose from outside the cell to inside the cell. So you can see here in our image, okay, permease, again, permease brings lactose from outside to inside the cell. Now, what are beta-galactosidases? Now, beta-galactosidases is an intracellular enzyme that cleaves the disaccharide lactose into glucose and galactose. Now, once lactose enters the cytoplasm of the bacteria, remember from the discussion on carbohydrates that lactose consists of two types of monosaccharides. We have glucose and galactose, right? So glucose and galactose, that is the component of lactose. Now that beta-galactosidase functions to cleave lactose into galactose to glucose. Okay. We have also transacetylase. So this transacetylase, These are enzymes transferring an acetyl group from acetyl-CoA to galactosides, lactosides, and glucosides. Okay. Now, glucose metabolized directly by glycolysis is the preferred energy source in E. coli. Now, other sugars can serve as the main source or main nutrient, but extra enzymatic steps are required to prepare them for entry into glycolysis, necessitating the synthesis of additional enzymes. So this is the structure of the lac operon. So we have the repressor gene, we have the promoter operator or the regulatory region, and we have the structural genes. So again, one by one, we have the repressor gene, the regulatory region. Inside the regulatory region, we have the promoter, the operator. For the structural genes, we have the LAC-Z, LAC-Y, and LAC-A. Again, for LAC-Z, LAC-Y, and LAC-A, it's ZYA. For LAC-Z, it codes the gene for galactosidase. For LAC-Y, it codes the gene for permease. For LAC-A, it codes the gene for transacetylase. Okay? What are the function of these components? So the operator, the LAC operator, this is the binding site for the repressor. So in the operator, it is again what I've previously mentioned. It is where the repressor is bound to in the LAC operon. The promoter, this is the binding site for RNA polymerase. So this is the binding where the RNA polymerase binding site. Again, in operator, it's where the repressor binds to. Where promoter, it is where the RNA polymerase binds to. Again, operator, repressor, in a promoter, it's RNA polymerase. Okay, for repressor, for this one, the repressor gene, okay, for this one, the repressor gene, it is the gene encoding the LAC repressor protein. So the repressor gene codes the repressor protein to bind it to the operator. Okay, so PI is the promoter for LAC-I, and CAP is the binding site for the CAP complex. So we have a more detailed picture of that. So we have again here the operator. The operator is where the repressor binds to. Promoters where the RNA polymerase bind to. CRE is the binding site for the CAP complex. Okay, so before we okay. So some operons are subject to positive control through stimulatory proteins such as the catabolite activator protein or CAP, which is an activator of transcription. Now, when glucose, which is a preferred food source of E. coli is scarce, CAP is activated by binding with cyclic AMP. Now, that activated CAP attaches to the promoter of the LAC operon and increases the affinity of RNA polymerase, thus accelerating transcription. Okay? The LAC operon contains genes again for the use of lactose. So I think this is just a reiteration. So in the presence of both glucose and lactose, bacterial cells prefer to use the glucose. Okay? It will not utilize the lactose if there's presence of glucose. So glucose prevents induction of the lac operon. How does it how does it prevent induction of lac operon? Through the binding of the CAP complex to the CAP binding site causes induction of lac operon. If you have high glucose levels, it causes low CAMP levels. But if you have therefore if you have a high glucose, you have a low CAMP, therefore you would have no induction of the lac operon. Again, in order for the lac operon to be induced or to be activated or to be expressed, you need to have this CAP-CAMP complex. Okay? This CAP-CAMP complex is required for the induction of lac operon. It requires for the expression of the lac operon. Now, if you have high glucose inhibits formation of CAMP. Okay, therefore causing low levels of CAMP. Therefore, if you have a low levels of CAMP, you won't form this cap-CAMP complex. If you have no cap-CAMP complex, you have no induction of the lac operon. Okay. So additional system, superimposed on repressor operator system allows capture of more energy from glucose. Okay, again, glucose is the preferred over lactose. And okay, just to emphasize again, if you have a high glucose, you have a low CAMP. You have a low glucose, you have a high CAMP. And you need to have a high CAMP required for lac operation activation. Why? Because CAMP binds to the catabolic activator protein. Okay, it physically, that CAMP is an allosteric effector of CAP, and CAP physically interacts with RNA polymerase to increase its affinity for the lac promoter. Again, the CAP or catabolite activator protein, therefore it's an activator. Activator of what? Activator of the RNA polymerase. In order for that CAP to be somewhat activated as well, you need to have a high CAMP. And in order for you to have a high CAMP or cyclic AMP, you need to have a low glucose inside the bacterial cell. Now, what happens to the expression of the lac operon when both glucose and lactose are present? So we have what we call catabolite repression. So catabolite repression restricts the expression of genes required for catabolism of lactose, arabinose, and other sugars in the presence of glucose. Why? Again, because of this reason. Even when these secondary sugars are also present. So the effect of glucose is again mediated by CAMP as a co-activator and an activator protein known as CAMP receptor protein or CRP, also known as CAP. So they are just the same. CRP or CAP, they are just the same. So it activates CRP or CAP, and that CAP activates RNA polymerase. So if you have a high glucose, it will inactivate adenylyl cyclase. Why is that? What is the function of this adenylate cyclase? That adenylate cyclase will form cyclic AMP. So if you have a high glucose, it inhibits the adenylate cyclase. If you have a low glucose, you have no inactivation. So it causes the conversion of ATP to CAMP or CAMP. That CAMP, when CAMP is present, it causes binding to CAP or catabolite activator protein. So when CAP and CAMP bonds together, it forms an activated form of CAP. Now that CAP binds to the RNA polymerase to activate the RNA polymerase. So this is the first circumstance. If you have a high glucose, therefore again, you have a low CAMP, but a lactose is absent, what happens is that when lactose is absent, the repressor binds to the operator and prevents transcription of lac genes. So if the bacteria contains the environment of bacteria has so much abundant of glucose, the repressor protein is bonded to the operator site. Okay. Okay. So the LAC-I or the gene that codes for the repressor is of course on, and that repressor binds to the operator site. So there will be no gene expression. Since you have no lactose and lactose binds to the repressor, okay, there will be no expression of genes for lac Z, Y, and A. What happens if glucose is low? Okay, the CAMP is high, but lactose is still absent. So for this one, the glucose is high. This one, the glucose is low, but the lactose is still absent. Since lactose is still absent, the repressor binds to the operator and binds the transcription of lac genes. Even if you have no glucose and at the same time you don't have lactose, the lac operon will remain off. You need to have lactose for that lac operon to be expressed. And why is that? Because the system wants to ensure that if it expresses that protein, that enzyme, that protein will be utilized by the cell, right? It's useless to make, for example, galactosidase if in the first place you don't have lactose. Remember, galactosidase converts lactose to glucose and galactose. So if your cell produces that enzyme to break down lactose, but in the first place you don't have lactose, so what's the point? So that's the main point. Even if you have whether the glucose is high or low, but if lactose is absent, there will be no gene expression of LAC-Z, Y, and A. Okay. Now, what happens if glucose is high? So therefore, CAMP is low, but lactose is present. So if lactose is present, the repressor dissociates from the operator. However, if glucose is also available, the low CAMP levels prevent the CRP-CAM formation and DNA binding. So remember that in order for the RNA polymerase to be activated, it requires, or it is better to have that activated form of CRP. Okay, so for this one, since you have high glucose, therefore you have a low CAMP, so that CAMP CRP complex will not form. So you have a low levels of gene expression, but there are still expression of rmRNA. Okay, so mRNA polymerase may occasionally bind and initiate transcription, but it results into a very low level of lack gene expression. What happens if you have a low glucose but you have lactose? So if you have low glucose and lactose is present, okay, what happens here is that again presence of CAMP. That CAMP will form a complex with CRP. If you have the CAMP CRP complex, it facilitates binding to RNA polymerase, forming or it causing activation of RNA polymerase. So therefore, you have a high levels of gene expression. Now, what happens to the repressor for this one? For these examples, for for instances where where lactose is quite abundant. So the presence of lactose causes binding to the repressor. And once lactose binds to the repressor, it causes dissociation. Okay, see this one? It causes dissociation of the repressor from the operator side, allowing the RNA polymerase to proceed to gene expression. Okay, the next type of system is the repressible system or the trip operon. So in contrast to that of the lac operon, which is an inducible system, the trip operon is a repressible system. So the system is initially on, transcription is ongoing and making a product. Now, operator can be turned off by a presence of a repressor, which is made active by being activated by a co-repressor molecule, which is usually the end product. An example of this one is the tryptophan operon. And the tryptophan acts as a co-repressor. Remember, tryptophan is not the main repressor, okay? Tryptophan is the co-repressor, inhibiting further synthesis of enzyme involved in the processes. Okay? So the trip operon is quite complicated. It requires a lot of genes to be remembered. But if you get the gist of it, what it means is that your the bacterial system makes a way not to over accumulate tryptophan. If you have enough supply of tryptophan, it needs to turn off the enzymes producing tryptophan. Okay, so this is the structure of the tryptophan. So we have the control sites just like in the lac operon. We have the promoter, the operator, and then we have also an attenuator. And you have these structural genes, the LEDCBA. The TRPE gene codes for antranilate synthase component 1. For TRPD, it codes for antranilate synthase component 2. For TRPC, it codes for the N5' phosphoribosyl antranilate isomerase indole 3 glycerol phosphate synthase for TRPB. it codes for tryptophan synthase beta subunit. For TRPA, codes for tryptophan synthase alpha subunit. Now, basically, the structural genes coding for the trip operon, what it does is that basically converts chorismate to form L-tryptophan. The chorismate is the precursor of L-tryptophan. Okay? So again, tryptophan metabolism in prokaryotes. So the genes for the enzymes needed to synthesize a given amino acids are generally clustered in an operon and are expressed whenever existing supplies of that amino acids are inadequate for cellular requirements. Now, once the amino acid, for example, tryptophan, is abundant, you have a lot of tryptophan, the biosynthetic enzymes are not needed. So the operon should be repressed because you don't want an overly supplied tryptophan, again because you want to maintain that balance. So the E. coli tryptophan operon includes 5 genes that I've previously described required to convert chorismate to tryptophan. The E. coli trip operon encodes 5 polypeptides, which form 3 enzymes that mediate synthesis of tryptophan from chorismate. So the 5 trip operon genes are coordinately expressed under control of trip repressor. Okay, we have a trip repressor which binds L-tryptophan to form a complex that specifically binds to the trip operator to reduce the rate of trip operon transcription 70-fold. So in this system, tryptophan acts as a co-repressor since its presence prevents what would superfluous tryptophan biosynthesis. Okay. So when tryptophan is abundant, the tryptophan binds to the trip repressor. Again, the excessive tryptophan binds to the trip repressor, causing a conformational change that permits the repressor to bind to the trip operator and inhibit the expression of the trip operon. The trip operator sites overlap the promoter, so binding of the repressor blocks the binding of RNA polymerase. So if you have a high levels of tryptophan, it causes termination of gene expression. Basically, that's the main point. If you have a low tryptophan, um, the RNA polymerase is not blocked by the repressor, so transcription will proceed. So I'll show you a picture of how the trip operon would look like.
The trip operon in E. coli contains 5 structural genes corresponding to enzymes that convert chorismate into tryptophan. The TRPE gene contains a 5' untranslated region, or UTR, that plays an important role in the regulatory mechanism called attenuation.
Okay, I'm sorry.
The 5' UTR contains 4 regions. Region 1 encodes a short peptide containing tryptophan amino acids. When tryptophan levels are high, region 3 pairs with region 4. This structure terminates or attenuates transcription. When tryptophan levels are low, region 2 pairs with region 3. This structure does not terminate transcription. Let's examine how these alternate structures arise. RNA polymerase begins transcribing the DNA. Closely following RNA polymerase, a ribosome begins to translate region 1. When tryptophan is abundant, the ribosome doesn't slow down at the tryptophan codons. By the time region 3 is transcribed, the ribosome has already moved past the tryptophan codons to partly cover region 2. Because region 3 is prevented from pairing with region 2, it pairs with region 4 instead to produce the attenuator, which terminates transcription. The structural genes are not transcribed, and therefore no additional tryptophan is synthesized. When tryptophan is scarce, the ribosome stalls at the tryptophan codons. Therefore, region 2 is not covered when region 3 is transcribed, allowing regions 2 and 3 to form a hairpin that does not terminate transcription. Because region 3 is paired with region 2, it cannot pair with region 4 and form an attenuator. Transcription continues. RNA polymerase transcribes the structural genes, which are translated into enzymes, which in turn synthesize more tryptophan. You can mimic the effect of tryptophan levels by determining how long the ribosome pauses at the trip codons.
Okay, so now let's move on to eukaryotic gene expression, eukaryotic gene regulation. So the level of gene regulation in eukaryotes, as what I've previously mentioned from the first slides, can be in the RNA transcription, nuclear RNA processing, mRNA stability, and translation. But in eukaryotes, levels of gene expression can also occur at the level of the DNA, even the level of the chromosomal structure. For eukaryotic transcription, general transcription factors binds to the promoter region of the gene, and RNA polymerase 2 then binds to the promoter region to begin transcription. To begin um, to begin transcription at the start site, which is the +1. Now, enhancers are DNA sequences to which specific transcription factors called activators bind to increase the rate of transcription. So we have promoters. That promoters binds some transcription factor that can activate RNA polymerase. And upstream, we have enhancers. These enhancers can bind to activators that conform and that can interact with the transcription factors, further stimulating the RNA polymerase to express genes. Now we have co-activators and mediators. They are also required for the function of transcription factors, and co-activators and mediators bind to the transcription factors and bind to other parts of the transcription apparatus. Okay, so activators, these are regulatory proteins that bind to DNA at a distant site known as the enhancer. So for this one, enhancers, what I've previously mentioned, enhancers are quite distant from this part. Okay, this one is also an enhancer. This is also an enhancer. Now, when DNA folds, so that enhancer is brought into proximity with initiation complex, the activator proteins interact with the complex to increase the rate of transcription. The co-activators, these transcription factors transmit signals from activators proteins to the general factors. Now, the general factors, these are transcription factors that position RNA polymerase at the start of a protein coding sequence and then release polymerase to initiate transcription. Now, the eukaryotic chromosome structure also plays a
Bacteria
role.
need to produce specific enzymes in order to meet its metabolic demand and also to reproduce.
No, the eukaryotic chromosome also plays a role in gene expression. Now, remember that eukaryotic DNA is packaged into chromatin. Okay? Chromatin structure is directly related to the control of gene expression. And chromatin structure begins with the organization of DNA to nucleosomes. And some nucleosomes and the organization of nucleosomes may block RNA polymerase from gaining access to promoters, thus making one way of regulating gene expression. Now, methylation or the addition of a methyl group of DNA or histone proteins is associated with control of gene expression, and cluster of methylated cytosine nucleotides bind to protein that prevents activators from binding to DNA. Now, methylated histone proteins are associated with inactive regions of the chromatin. Now, we have also the histone covalent modification. Now, the following are the possible roles of post-translationally modified histones. Okay, again, post-translationally modified histones. The acetylation of histone H3 and H4 is associated with activation or inactivation or gene expression. Okay, acetylation of core histone is associated with the chromosomal assembly during replication. For phosphorylation of histone H1, it is associated with the condensation of chromosomes during replication cycle. The ADP ribosylation of histone is associated with DNA repair. For methylation of histone is correlated with activation and repression of gene transcription. Now, monoubiquitination is associated with gene activation, repression, and heterochromatic gene silencing, and summoylation of histone, or SUMO, what SUMO stands for small ubiquitin related modifier, is associated with transcription repression. Now, certain DNA elements enhance or repress transcription of eukaryotic genes. So we have enhancers or silencers. Now, enhancers, they are certain DNA elements that facilitate or enhance initiation at the promoter. And silencers, these are cis-acting DNA elements that decrease the expression of specific genes. One of the best understood mammalian enhancer system is the beta interferon gene. Now, what are the properties of enhancers? They work when located long distance from the promoter. They work when upstream or downstream from the promoter. They work when oriented in either direction and can work with homologous or heterologous promoters. They can work by binding one or more proteins. They work by recruiting chromatin modifying co-regulatory complexes. They work by facilitating binding or function of the basal transcription complex at a cis-link promoter. For post-transcriptional regulation, control of gene expression usually involves control of transcription initiation, but gene expression can also be controlled once the RNA is formed. So we have mechanisms such as RNA interference, alternative splicing, RNA editing, and mRNA degradation. For RNA interference, it involves the use of small RNA molecules. The enzyme dicer chops the double-stranded RNA into small pieces of RNA. So we have microRNAs bind to complementary RNA to prevent translation. We have also small interfering RNAs to degrade particular mRNA before translation. Okay, so they prevent translation. They are already transcribed, but the process of before translation is where this post-transcriptional regulation takes place. Now, introns also are spliced out of pre-mRNA to produce mature mRNA that is translated, and alternative splicing recognizes different splice sites in different tissue types. So the mature mRNAs in each tissues possesses different exons, resulting in different polypeptide products from the same gene. So this alternative splicing causes variation of the gene product, but having the same gene. Okay, this is what happens during splicing. Okay, so the introns are spliced. Okay, they are somewhat removed. Okay, and the pattern of removal of these introns creates the different types of patterns of the splicing causes production of a unique mature mRNA. Okay, so for example, for this one, for when there's when there's an intron splicing from original primary RNA transcript, if it's towards the thyroid splicing pattern, you would see that the exons that are found in this part are only 1, 2, and 3, and 4 exons, while for the CGRP, you have 1, 2, 3, 5, 6 exons forming CGRP, and this is calcitonin. And the reason why there's a different pattern of exons or the different arrangement of the exons because of the how the introns are spliced. Now, RNA editing creates mature mRNA that are not truly encoded by the genome. So, for example, we have the apolipoprotein It exists in two isoform. One isoform is produced by editing the mRNA to create a stop codon, and this RNA editing is tissue specific. Now we have also mature mRNA molecules that have various half-lives depending on the gene and the location of expression. The amount of polypeptide produced from a particular gene can be influenced by the half-life of the mRNA molecules. And mRNA stability is subject to regulation. Now, we have also protein degradation. Now, once translation has already occurred, okay, proteins are can be degraded continually inside the cell. And proteins can be degraded and tagged by ubiquitin, and degradation of proteins is marked by ubiquitin occurs at a proteasome. Okay, so in order for a protein to be destroyed, it needs to have tagging by ubiquitin, and once ubiquitin is bound to the protein, it is sent to the proteasome, wherein that protein that already underwent ubiquitination will be degraded to form polypeptide fragments and thus be recycled by the human body. Okay? So thank you very much for your kind attention and study well on the regulation of gene expression, and please read the references materials that I provided in the first few slides of this lecture. Thank you so much and have a safe day.